Welcome to the BiochemSerye episode entitled Monosaccharides. This is an episode of the playlist on chemistry of carbohydrates, which is linked above. Feel free to click above and visit the playlist to view the other episodes on this topic. Let us now discuss a very important segment of carbohydrates, monosaccharides, the building blocks of polysaccharides, and other important sugars. Only the most important of a large number of naturally occurring monosaccharides are biomedically important. As mentioned in the structure of carbohydrates, they are classified according to the number of C atoms, into pentoses, hexoses, etc., and according to the chemical nature of the carbonyl function into aldoses and ketoses. Let us start with the aldoses, and with the 5-carbon aldoses, the pentoses. The best-known aldopentose, D-ribose, is a component of RNA and of nucleotide coenzymes, and is widely distributed. In these compounds, ribose always exists in the furanose form. Like ribose, D-xylose and L-arabinose are rarely found in free form. However, large amounts of both sugars are found as constituents of polysaccharides in the walls of plant cells. The most important of the aldohexoses is D-glucose. A substantial proportion of the biomass is accounted for by glucose polymers. Above all, cellulose and starch. Free D-glucose is found in plant juices, for example, grape sugar, and as blood sugar, found in the blood of higher animals. A C4 epimer of glucose, Dgalactose, is a constituent of lactose or milk sugar. It is found in glycolipids and glycoproteins. D-mannose, a C2 epimer of glucose, is important in the glycosylation of certain proteins. Let's proceed to the ketoses. The best examples of the ketoses biomedically are the ketopentoses. Phosphoric acid esters of the ketopentose D-ribulose are intermediates in the pentose phosphate pathway, and in photosynthesis. The most widely distributed of the ketohexoses is D-fructose. In free form, it is present in fruit juices and in honey. Bound fructose, on the other hand, is found in sucrose and plant polysaccharides, like inulin. Moving on, let's look at aldoses wherein one or more of the O groups are reduced at certain carbon. They are known as deoxyldoses. In deoxyldoses, an OH group is replaced by a hydrogen atom. 2-deoxy-D-ribose is reduced at C2 and is known significantly as a component of DNA. Fucose is a hexose deoxy sugar that is reduced at C6. It is found on N-linked glycans on the mammalian, insect, and plant cell surface. In organic chemistry, an amino sugar, or more technically a 2-amino-2-deoxysugar, is a sugar molecule in which a hydroxyl group has been replaced with an amine group. When this amine group is acetylated, it then becomes an acetylated amino sugar. The acetylated amino sugars N-acetyl D-glucosamine and N-acetyl D-galactosamine are often encountered as components of glycoproteins. More than 60 amino sugars are known, with one of the most abundant being N-acetyl D-glucosamine, which is the main component of chitin. The sugar acids are obtained when a carbonyl group or a hydroxyl group is oxidized to a carboxylic acid group. Here are several examples. N-acetylnuraminic acid, more commonly known as sialic acid, is a characteristic component of glycoproteins. Other acidic monosaccharides such as D-glucuronic acid, D-galacturonic acid, and liguronic acid, are typical constituents of the glycosaminoglycans found in connective tissue. Sugar alcohols, also called polyhydric alcohols, polyalcohols, aldehydes, or glycidyls, are organic compounds, typically derived from sugars, containing one hydroxyl group attached to each carbon atom. Sorbitol, a sugar alcohol, is formed by the reduction of the glucose aldehyde group. Sorbitol is significant in diabetes mellitus and the pathology of cataracts. If the sugar were mannose, the resulting sugar alcohol would be mannitol, a known osmotic diuretic. This concludes this episode of the BiochemSerye series of this topic. Feel free to watch the other BiochemSerye episodes of this lecture as linked on the next screen, and in the description below. Please subscribe to our channel, the BiochemSerye channel, click on the notification bell button, and be notified of new videos to be uploaded. Content will be added regularly. Thank you for watching this BiochemSerye episode. You can check out the related videos linked here or click on the related playlist as shown to view all the videos of this topic.